Good morning, children. So today I am going to start a new chapter. The name of the chapter is photosynthesis in higher plants. The word photosynthesis consists of two words, photo and synthesis. Photo means light and synthesis. Synthesis means to make. What it is making? It is making organic substances that may be proteins or carbohydrates or fat or vitamin or nucleic acid, DNA or RNA or hormones or any other substance. In fact, all the organic substances which we see around us, they all are ultimately derived from the substances which are produced by the plant. May it be plastic, may it be paper, may it be wood, may it be anything else for that sake. Even the synthetic shirts which we are wearing and the trousers and all, okay, which is ultimately made up of nylon or rayon or polyester or whatever it is, and they are all using some of the organic substances which we may be getting from the petroleum, and petroleum was actually formed by the decomposition of the organic matter of the past, which is actually produced by photosynthesis. So we can say that the photosynthesis is not only providing us food, but it is also providing some of the basic necessities for us. So it is a very important process. The process of photosynthesis is producing at least food, and it is also a process in which the oxygen is produced. These are the two vital things without which we cannot even imagine the survival of any other organism. Okay, even those which are inorganic in nature, they are also depending on the organic substance. Those which are anaerobic in nature, they are also at least depending on the food. Okay, they may not need oxygen, but at least they are needed. So for the very survival of any organism, we are ultimately thankful to the plants. Now, the process of photosynthesis is known to us for years. Okay. So there is a history. If you want to know, that is of course the reaction which we write today, that six carbon dioxide plus 6 molecule of water gives 1 molecule of glucose plus 6 molecule of oxygen. That has given very long back. Okay. But in due course of time, there are several developments took place in that. Here some of the historical events are given, like early experiments. It was Joseph Priestley who has taken one bell jar in which he kept a rat and a candle, and both the rat also died and the candle also died. In the same bell jar, when he placed a plant, the candle also continued to burn and the rat also continued to survive. And on the basis of this experiment, he said that is the living beings and the candle actually fouls the air, whereas the plants which purify air. Of course, he was not knowing anything about oxygen. However, later on, he only was the discoverer of oxygen, and he did a lot of study on the oxygen. And then many other experiments are conducted one after another. That is another experiment which is given. Now consider the interesting experiment done by the T.W. Engelmann. Now what is Engelmann is that is using the prism of the light to split into the 7 colors. Then the cladophora, it's a type of algae, it's a type of micro phytoplankton, okay, aerobic bacteria, and they were used to detect the site of oxygen evolution and observed that the bacteria accumulate mainly in the region of the blue and the red light. When the photosynthesis is taking place, actually it is consuming or it is producing the maximum oxygen evolution is taking place in the blue light and the red light. So all these cladophora used to go and accumulate there. In the other lights, that is the green light, yellow light, orange light, and the other lights, where they were producing oxygen merely in a very fraction. So on the basis of this reaction, this observation in the 19th century, they came to know that the carbon dioxide and water is actually responsible for production of some of the organic substance. This is actually empirical formula CH2O, whereas the value of C and the value of O is same and H is double to that. So if we place 6, so it will be C6H12O6. If it is 3, to C3H12O3. Okay? Like that it is. So it is an empirical formula which was given on the basis of their certain calculation. Okay? The milestone contribution of understanding the photosynthesis was made by microbiologist Cornelis von Neal, who based on his studies on the purple and the green bacteria demonstrated the photosynthesis is essentially a light dependent reaction on which hydrogen from a suitable oxidizable subcompound reduces the carbon dioxide into carbohydrates. So that was one of the great contribution. And now in this reaction, if you see H2A, now this A is actually, it can be O or it can be S. So There are certain organisms, microorganisms, as I just said, that they are in place of water, they are using hydrogen sulfide. So when they use hydrogen sulfide, they will be giving out sulfur. And all the higher plants which we see around us, they all are using water. So oxygen is given by the oxygen will be given out as a waste material or as a byproduct, and the hydrogen from the water is actually used for the reduction of carbon dioxide. So then the reaction was given like this, the photosynthesis reaction, that is C6 carbon dioxide plus 6 H2O gives 1 molecule of glucose plus 6 molecule of oxygen. However, later on it was found that the oxygen evolved. Okay, if I write this reaction, 6 carbon dioxide plus 6 molecule of water give me 1 molecule of C6H12O6 plus 6 molecule of oxygen. Now the question is, what is the source of this oxygen? Whether this oxygen has come from here or whether it has come from here. Obviously, we can see that is the 6O2 here, so 6O2 here. So the quite obvious source of the photosynthesis seems to be carbon dioxide. But when an experiment was conducted in which radioactive labeled hydrogen was taken, oxygen was taken, here water ICM plant is taken, and water and test tube को लगाया जाएगा इसके ऊपर, थोड़ा सा गलत हो गया ये, test tube थोड़ी सी लगाना है. So this is the glass funnel, and this is a test tube filled with water, and this is the water. Now here the water was taken. The water was taken was H2O18 in place of O16. Okay. So when the photosynthesis was done, what was found that the oxygen evolved was oxygen 18. So that means the very source of oxygen which is evolved is actually water, not the carbon dioxide. So the source of this water should be oxygen from water. So then we'll have to change the reaction, we'll have to rewrite the reaction, we'll have to make certain changes in our original belief. The 6 carbon dioxide will be there, no problem. But in place of 6 H2O, now it will be 12 H2O. Then only the number of oxygen will be 12, which will be equivalent to there. So C6 H12O6 plus 6 O2 plus 6 H2O. So now to make this water, the oxygen comes from carbon dioxide. So this is how the belief has to be changed. Okay, so now today we know that the source of oxygen which is evolved in the photosynthesis is actually water, not the carbon dioxide. Okay, so these are the earlier development. Now, where does the photosynthesis take place? It's quite obvious. The photosynthesis is taking place in chloroplast. Okay. If I explain it in simple words, I can say that there are 4 basic necessities of the photosynthesis. What are the basic necessities of photosynthesis? Photo, that is the light, then carbon dioxide, then water, and then chloroplast. Let us highlight on one of them, all of them one by one. Okay. So 4 things are there, that is and light. Let us talk about the carbon dioxide. The source of carbon dioxide for the land plant is air, and for the aquatic plant it is water. In the air it is available in the form of 0.03%, and in water it is available up to 3%. Okay? In water. And this carbon dioxide is actually taken in by stomata. In the aquatic plants it diffuses through the stomata and through the other organ also where the cuticle is not formed. So there it will be taken in and this. So carbon dioxide is not a big problem. Nowadays we know that most of the industrialization is taking place, amount of pollution is taking place, the amount of carbon dioxide is increasing. But the thing of worry is that the number of plants are decreasing. Had we produced enough of plant, had the plants were enough, this carbon dioxide would have been converted back into the oxygen, and all the greenhouse effect and all other things, whatever type of problems which we are facing today would have not been there. But we human beings, we simply cut down the trees non-judiciously. That is the problem. Now what is the next thing is, that is water. water is the source of hydrogen for reduction of carbon dioxide. Water का source क्या है? Water is taken from the soil for all the land plant, and water is taken from the surrounding for all aquatic plants. ठीक है? So that is also not a big problem. Water actually, what happens? There is a process called splitting of the water or photolysis. In the photolysis, the 4 molecules, suppose we take 4 molecules of water, so it will break into 4 OH ions plus 4 H ions. H ions, as you know, very active, so they will be immediately taken by NADP, which will change into NADPH, NADPH plus H ion. Okay, and the 4 molecules of OH will rearrange to make 2 molecules of water plus 1 molecule of oxygen, which is given out as a waste product. So this is a process called photolysis. Photolysis is taking place in the chloroplast in the presence of light. Okay, to carry out this particular process in the laboratory, we require electric energy. We can split the water only using electricity. There is no other way. Whereas the plants are simply doing it in the presence of light in their chloroplast. So that is what I say, that the God is great. He knew several methods. This NADH2, that will be used actually for the reduction of carbon dioxide, so which we will study little later. Now come to the next thing, that is the chloroplast. Where the chloroplast is found? Chloroplast is found in the chlorophyll. Chlorophyll we have studied with the double membrane structure. It has got their own DNA. It has got some granathylakoid, and in the granathylakoid they got these pigments: chlorophyll A, chlorophyll B, xanthophylls, keratinoids, etc., etc. Okay, these are the plates which are connecting them. Okay, and this is called stroma. These are granathylakoid. And in these there are these pigments embedded. Now, those pigments which are there, they are chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, and even chlorophyll E. Besides this, there are xanthophylls, then keratinoids. So these are the pigments. Most importantly, photosynthesis require these two pigments. Chlorophyll A is must. Chlorophyll A is must, as chlorophyll A is actually a reaction center. So photosynthesis cannot take place without chlorophyll A. The other pigments are merely absorbing certain portion of light and passing it to the chlorophyll A. Okay. Now chlorophyll A is actually using red portion of the light, sunlight, and chlorophyll B is actually using the violet portion of the light. Since these type of chlorophylls are maximum available in the plants, so the maximum photosynthesis is taking place in these two places only. The other pigments are only found in a fractions. They are very small in amount. We have already studied about chlorophyll C, chlorophyll B, and chlorophyll A when we talked about, or when we studied about algae. Okay, which had a different combination, chlorophyll A and B, chlorophyll A and C and all. Okay, and the other pigments are also there. Then xanthophylls are there, which are giving them yellowish color, and the keratinoids are there, which are giving them orangish color. So you see there are different varieties of the leaves. No two plants leaves are of same color. So these are because of the different combinations of these pigments present in them. And plus this, you might have even seen there are some plants, there are some leaves which are beside being red, they may have some yellow spots, or they may have some red spots, etc. So that is because of the different combinations of these present in them. Of course, plastids are plastids, so they can interchange. Okay, so this is what the chlorophyll is. Now the next is light. Light, as you know, is there are different sources of the light. Maybe sunlight, maybe tube light, maybe candle light, maybe bulb light, maybe light from any source, and any light can be used in photosynthesis. We are not specifying that solar synthesis. We are not written in the solar light synthesis. We are not writing the sunlight synthesis. We are not writing the candle light synthesis. We are not specified what light. The source of light can be anything. Okay. Now, when we talk about the white light is actually split of several colors, and just for the convenience, Still, I just want to make it sure that the light has got infinite color. White light is containing infinite color. If you just go to any of your computer, what you call, any of the photo editor, you can see how many colors are there. Okay, all the hue setting which you do, orange to red or red to orange or to green, you see how many colors are there. So there are millions of colors, but just for the sake of our understanding, we have classified them in the 7 colors: VIB, G, G, Y, O and R. So the maximum photosynthesis are taking place at these two brackets. The minimum photosynthesis are taking place in this. If we plot a graph, so what we will find that the maximum photosynthesis is taking place in this region, and this yellow and green region it will decrease, and then again it will increase. Okay? However, again you will find that the maximum photosynthesis in the violet color. So this is because of the chlorophyll B, and this is because of the chlorophyll A. So there are two terms which is said, that is the action potential and not action potential, it is absorption spectrum and action spectrum. That is which portion of the light is absorbed maximum and which portion, in which spectrum the maximum photosynthesis is taking place. So it is directly related. Where more light is absorbed, more photosynthesis is taking place. Where less light is absorbed, less photosynthesis. Green light is, most of it is actually reflected. Because of that only we see the leaves as green in color. So naturally their role in the photosynthesis will also be minimum. It is quite obvious. You all are actually of class 11, so you all know all these things very well. So this is action spectrum and absorption spectrum, both are depending on one another. That is how much light will be absorbed, that much only photosynthesis will take place. And what is maximum photosynthesis taking place in? These two lights are absorbed in the maximum quantity, other lights are absorbed in the less quantity. Okay? So a tube light, a white light is consist of all these colors, so accordingly the photosynthesis will take place. Okay, so these are the basic necessity, basic understanding which we had. You can see here that chlorophyll A, carotenoids and chlorophyll B, which is given here on the page number 210. And you can see that is where the photosynthesis. So chlorophyll B is carrying the maximum photosynthesis in the violet zone. Okay, blue and violet zone. Whereas the chlorophyll A is carrying out maximum photosynthesis in the red light zone. Okay, the second diagram which is there, the rate of photosynthesis is measured by the oxygen release. So it is directly indication that is how much photosynthesis is taking place with the shown in the black light. And the last one is light absorbed. This is absorption spectrum. The absorption spectrum is saying that where the more absorption is taking place, where there only the maximum photosynthesis also took place. So the maximum light is absorbed towards the 400 nanometer and then towards 700 nanometer. In the 500 to 600 or 650 nanometer, the least light is absorbed, and that is why the photosynthesis is also least. Now come to the thing. Now photosynthesis, as we are seeing, it is not in the same way. It is not that the photosynthesis always takes place in presence of light. Okay, this is our misinterpretation. Light is actually required to make energy and to make energy. So the process of photosynthesis is divisible into two parts: the light reaction and dark reaction. Now dark reaction is not that which is taking place in the night. A light reaction is of course taking place in presence of light. Okay, it is something like that. Suppose you got two taps in your house, in your school campus. One tap is here, in which salty water comes. तो इसे क्या रहेगा? खराब नहीं है. This is salty water. ठीक है? So in the other tap where the water is consumable, which is water is potable, which is good in taste, so you will not say that this is a soft water or you will say, you will say this is sweet water. ये कोई स्वीट तो है नहीं, इसमें कोई शुगर तो है नहीं. It is our interpretation in that way. If it is salty, it is sweet. If it is dark, it is light. If it is light, it is dark. ठीक है. So that is why we say that way it is not that the light reaction require light or heat, but dark reaction require dark. ऐसा कुछ नहीं है. Dark reaction is also taking place in presence of light only during the daytime also. However, it can take place even in the night. Light reaction में दो products बनते हैं. क्या? The two products which are required, that is the energy and NADPH plus H ions. Or we can write it with NADPH2. Okay, these are the hydrogen donors. So during the photosynthesis just now, if you remember when I said the water molecule is split into two parts, that is the OH ion and H ion, and OH ion will recombine to make water and oxygen. The oxygen will be given out, and the H ions which are released, the H ions are actually absorbed by them. Okay. Also, the plant has an ability to synthesize ATP in presence of light. So these two things are taking place, that is always done in presence of light. And the second part, that is the reduction of carbon dioxide into organic substance, maybe C3H6O3 or C6H12O6 or starch, whatever you say. First this product is made, that product, and then later on it can be changed into starch. Okay, so this is taking place in the plant which is independent of light. If light is there, well and good. If light is not there, then also it will be done. Okay, so this is light independent process and this is light dependent process. Okay, now light dependent process itself is of two types. Light dependent process itself is of two types, that is called cyclic and non-cyclic. Cyclic and non-cyclic path. Okay, so let us study the cyclic and non-cyclic path, which is given here. Okay, on the page number 211. Okay, if you see the first diagram 13.4, what is there? That the light is falling. A photon, it is actually showing in the other direction. नीचे से ऊपर जा रहा है. Light actually, photons are falling onto the chloroplast. So this green color are the pigments. Pigments are actually chloroplast. Variety of chloroplast is there. They will accept the light, and when the light is falling into them, some of the electrons will charge and they will leave it. When these charged electrons are released, they are accepted by the primary acceptor, and after some time they will lose their energy and come back to the original molecule. So there is a cyclic part. निकलना है. It is something like that. एक बच्चा है, उसके घर में थोड़ी लॉटरी लग गई, उसको कुछ 1000-2000 रुपये मिल गए, तो वो क्या करेगा? That money will take, he will enjoy, he will come back. मनी 2-5-10,000, 20,000, 1,00,000 रुपये मिल गए, कितने दिन मौज करेगा? 1,00,000 रुपये खर्च होने में 5 दिन भी नहीं लगेंगे, डिपेंडिंग ऑन द नेचर ऑफ द चाइल्ड. फ्री का मिला है तो जल्दी खर्च होगा. ठीक है. सो यू विल एंड अल्टीमेटली कम बैक होम. सिमिलरली इन द लाइट रिएक्शन, इफ यू सी दिस डायग्राम, this is the diagram 13.6, figure number 13.6. you see the light wavelength are falling on the chlorophyll. chlorophyll कौन सा वाला? chlorophyll दो तरह के होते हैं. ये वाले chlorophyll कौन से हैं? इसको हम कहते हैं PS1. okay, now PS1 actually photosystem 1 is absorbing the wavelength of 700 nanometer. So of the light, only 700 nanometer light will be absorbed, and some of the electrons will become charged and they will leave the chlorophyll, and then there are primary acceptor, they will accept it. So that it's shown there, electron acceptor. Okay, of course it is a long process, but in your book it is summarized, and कोरोना काल में तो आप लोग वैसे ही, ठीक है, छोड़िए. तो प्राइमरी एक्सेप्टर ने इसको एक्सेप्ट करा, उसके बाद दैट हैज पास टू दैट सेम इलेक्ट्रॉन ट्रांसपोर्ट चेन एज वी हैव स्टडीड इन यस्टरडेज क्लास, दैट इज इन द इलेक्ट्रॉन ट्रांसपोर्ट चेन इन रेस्पिरेशन. सिमिलरली इट इज पासिंग थ्रू द 1 to 2nd, 2nd to 3rd, 3rd to 4th, and during that course enough of energy is released that can be used to convert 1 ADP into 1 ATP. ठीक है. 2 photons energy will be used to convert 1 ADP into 1 ATP. Figure you can see 13.6. Very simple, कोई चौंसला ही नहीं है. ठीक है. So in the cyclic path of phosphorylation is actually standing for recharging of ATP. ADP plus phosphate is ATP, that is phosphorylation. तो if लाइट की प्रेजेंस में हो रहा है इसलिए इसको फोटोफॉस्फोराइलेशन कह दिया। और इस प्रोसेस के अंदर क्या है, कोई झंझट नहीं है, सनलाइट आई, एनर्जी निकली, दैट एनर्जी इज कन्वर्टेड इनटू ATP. We shall study that second type of change here, that is called Z scheme. Now I don't know why they are given name Z scheme. They could have even given it as the N scheme. चलिए, just pay attention. Now in the non-cyclic path, the light photon are falling onto the two pigment LHC1, LHC2, light harvesting chlorophyll. लाइट हार्वेस्टिंग क्लोरोफिल दो तरह के हैं, एक है P2 फोटोसिस्टम 2. फोटोसिस्टम 2 इज एब्जॉर्बिंग 680 नैनोमीटर लाइट एंड PHC दैट इज फोटोसिस्टम 1 इज एब्जॉर्बिंग 700 नैनोमीटर लाइट. सो व्हाट हैपेंस, वेरी सिंपल सा प्रोसेस यहाँ पे लिखा है, इसको मैं यहाँ पे थोड़ा सा ड्रॉ कर देता हूँ जस्ट फॉर द सेक ऑफ अंडरस्टैंडिंग. P680, P680 इस पे फोटोन गिरे, दैट रिलीज इलेक्ट्रॉन्स. दीज इलेक्ट्रॉन्स आर एक्सेप्टेड बाय द प्राइमरी एक्सेप्टर. प्राइमरी एक्सेप्टर से ये निकलेंगे, निकलने के बाद दे विल नॉट कम बैक टू P680. दे आर नॉट रिटर्निंग. इनको इतनी शर्म आती है क्योंकि ये घर छोड़ गए तो दोबारा अपने पापा के घर पे नहीं आते, ये किसी और के घर में जाकर के रहना पसंद करेंगे. तो दे कम टू P700. P700 also received 2 photons. उनके भी इलेक्ट्रॉन छोड़ के चले गए थे, क्योंकि उनके इलेक्ट्रॉन छोड़ गए थे तो these electron when come back to they receive them, they accept. बोले हमारे क्या, हमारा बेटा गया, हमारे को दूसरे का बेटा मिल गया, बात खत्म. ठीक है. सो इनका जो बच्चा चला गया है तो इसको तो दुख होगा ना, ये लॉस्ट हिज चाइल्ड, सो इट इज अ पेनफुल सिचुएशन फॉर हिम एंड हिज चाइल्ड इज सो शेमफुल, so ashamed that he is not returning to his own home. He knows that he has said so many things to his daddy and he left the house, so he is not coming back. During this change, that had enough of energy which used to convert one ADP and phosphate into ATP. इसी कहानी तो बहुत अच्छी है पर मेरे पास अभी समय नहीं है, तो मैं भी ज़्यादा नहीं पूछ सकते हैं, बट इट इज़ वेरी इंटरेस्टिंग टू अंडरस्टैंड. ओके, अब इसका इलेक्ट्रॉन जो है फिर वो दूसरा, इसका जो इलेक्ट्रॉन निकला था वो इसके पास चला गया, एक्सेप्टर के पास, प्राइमरी एक्सेप्टर. ये ऑल्सो प्राइमरी एक्सेप्टर. और यहाँ पे बैठे हैं NADP. ओके, नाउ कब फोटोलाइसिस का वो जो प्रोसेस मैंने वहां पे बताया था वो एक्चुअली यहाँ हो रहा है, फोटोलाइसिस. सो H2O, द H2O विल स्प्लिट इनटू H आयंस. तो दो लिख देते हैं इसको, ठीक है. सो दे नहीं, एक काफी, तो 2 H आयन एंड हाफ मॉलिक्यूल ऑफ ऑक्सीजन प्लस हाफ मॉलिक्यूल ऑफ ऑक्सीजन. तो हाफ मॉलिक्यूल ऑफ ऑक्सीजन विल बी गिवन आउट एंड दीस H आयंस विल बी सेंड हियर एंड इसमें से 2 इलेक्ट्रॉन भी निकल रहे हैं, वो 2 इलेक्ट्रॉन चले गए यहाँ. भाई H थे ना, हाइड्रोजन थे, हाइड्रोजन में से इलेक्ट्रॉन यहाँ चले गए, प्रोटॉन वहाँ चले गए और वाटर को इन्होंने ऑक्सीजन को धक्का मार के बाहर निकाल दिया. सो द प्रोसेस ऑफ फोटोलाइसिस इज टेकिंग प्लेस हियर. तो अब पूरे प्रोसेस को समझ लेते हैं एक बार दोबारा. P680 के ऊपर लाइट गिरी है एंड इसके 2 इलेक्ट्रॉन, इट इज समथिंग लाइक दैट कि दे गॉट लॉटरी एंड दे लेफ्ट द होम. 2 ब्रदर्स कहा चलो यार यहाँ रहेंगे तो पापा मम्मी को टीवी खरीद के देना पड़ेगा, ये रहेगा. सो दे जस्ट लीव द हाउस एंड गो टू प्राइमरी एक्सेप्टर. ये बड़ा होटल है, इस होटल से फिर थोड़ा छोटा होटल, तो दे आर स्पेंडिंग ऑल देयर मनी. ड्यूरिंग दिस कोर्स दे हैड इन्वेस्टेड सम मनी, कुछ का उसने गोल्ड पर खरीदा, बट दे फील सो अशेम्ड टू रिटर्न होम कि वो वापस तो नहीं आएंगे. अब जब पैसा खत्म हो गया तो ये कम बैक, बट दे लैंडेड टू सम अदर हाउस. उसके भी बच्चे निकल गए थे, वो चले गए वहां पर, और उसके बच्चे जो है वो भी वापस लौट के नहीं आए. On the other hand, water के molecule के ऊपर भी लाइट गिरी, that is split into H ions and oxygen, half molecule of oxygen. और इसमें से 2 इलेक्ट्रॉन निकलेंगे, तो इसका बच्चा निकला, उसकी भरपाई यहाँ से होगी, इसका बच्चा निकला, उसकी भरपाई यहाँ से होगी, इसका भी तो निकला है, ये कहाँ जाएगा? सो यहाँ 2 एचाइंस, 2 इलेक्ट्रॉन एंड 1 NADP। NADP इसको और इसको ले लेता है, चेंज कर देता है, NADPH plus H। Beautifully finished. So this is called non-cyclic path. So if you say the difference, the cyclic path and non-cyclic path, in for cyclic path P700 used, non-cyclic path P700 and 680 both are used. In cyclic path only ATP is produced, their ATP and NADP both are produced. Here oxygen is not released, their oxygen is released. Here NADP is not formed, their NADPH2 are formed. ठीक है, here photolysis is not taking place, here photolysis is taking place. So these are the things. Okay, so ये हो गया हमारा काम। हम आगे the cyclic and non-cyclic मैंने पढ़ा दिया। Now we can come the chemosynthetic hypothesis. Now chemosynthetic hypothesis is same as we are done in respiration. क्या हो रहा है देखिए, where is this process of photosynthesis, this splitting of water taking place? Where is this splitting of water taking place? Splitting of water is taking place inside, यहाँ पे हो रहे हैं, यहाँ पे inside the grana thylakoid, and as a result there will be lot of H-ion produced there. ठीक है, now where these H-ions will go? These H-ions want to come out, क्योंकि वो तो gradient create हो गया ना, अंदर ज्यादा H-ion है, बाहर H-ion कम है, so the gradient will allow them to go, and where this come out from the F0F1 particle. And we have studied in yesterday's class, when one pair of H-ions comes out, that has enough of energy to convert one molecule of ADP into ATP. So this is what the process is. So you see here P600 here, P680 ये रहा और P700 ये रहा, इसके इलेक्ट्रॉन जो है यहाँ तक आ गए। अब ये इलेक्ट्रॉन जो है वो हाइड्रोजन आयन इलेक्ट्रॉन मिलके NADPH बना दिए, है ना, एंड ये हो गया, और ये H-ion जो यहाँ से निकल कर आ रहे थे वो वहाँ पे यूज हो जाएंगे, एंड इनसाइड दैट वॉट एवर H-ions वर देयर, दीज H-ions जो हैं वो इधर से बाहर निकलेंगे, तो यार ATP सिंथेसिस विल फॉर्म। ठीक है, सो इट इज अ वेरी सिंपल प्रोसेस। व्हाट इफ यू रिमेंबर व्हाट वी आर स्टडीड इन यस्टरडेज क्लास, टुडेज क्लास विल बी इजी। ठीक है, सो दैट्स ऑल। अब हम आते हैं द नेक्स्ट प्रोसेस, दैट इज where the ATP and NADPH2 are used, where NADPH2 and ATP are used. So I said that photosynthesis is divisible into 2 phases, light phase, light reaction, and the dark reaction. And again and again and again and again I emphasize, the dark reaction is not a reaction which is taking place in the night during dark. Dark reaction is very well taking place during daytime only, simultaneously along with the light reaction. But it is called dark reaction because in this dark reaction light is not necessary. लाइट नहीं हो तब भी चलेगा, लाइट की प्रेजेंस में ATP वो बन गए, बनने का प्रोसेस वहां हो गया, अब उनको कंज्यूम करना है। तो ATP and NADPH2 बनाने के लिए लाइट चाहिए थी, दैट इज टू मेक द ATP and NADPH2 energy was required, light energy was required. For their reduction there is no energy required, energy whatever energy is required that will be provided by ATP which is produced during light reaction. Is it clear? Now this dark reaction is named in the different ways. You can call it the dark reaction, you can call it the Kelvin cycle, Melvin-Kelvin cycle, you can even call it the what you call TCA cycle, है ना, तो इसके अलग अलग नाम दे रखे हैं, है ना। चलिए हम बात करते हैं दिस Kelvin cycle, गेट Kelvin cycle। वेरी सिंपली इट इज गिवन, आपके बुक में तो कोई चौंचल ही नहीं है। इन द प्रीवियस कोर्स इट वाज सो बिग एंड सो मच एक्सटेंड कर देयर, नाउ दे आर सिंपली मेड इट वेरी सिंपल, एंड दैट इज व्हाई द चैप्टर आल्सो गोज इन अ वेरी इजी शॉर्ट। खत्म ही हो गया चैप्टर मान लीजिए, अगर आपने Kelvin रिएक्शन पढ़ा दिया आज तो फिर बचा ही क्या। चलिए साहब यहाँ पढ़ते हैं, हमारे पास एक मॉलिक्यूल है, इसका नाम है RUBP, रिबुलोज बाइफॉस्फेट, रिबुलोज बाइफॉस्फेट। इसमें 5 कार्बन हैं साहब, 1 2 3 4 5, इसके हमने लिए 6 मॉलिक्यूल। तो हाउ मेनी टोटल कार्बन्स आर देयर, 6 into 5 दैट इज 30 कार्बन्स आर there. This ribulose biphosphate यहाँ है एंड इसमें 6 मॉलिक्यूल कार्बन डाइऑक्साइड के लिए. So each molecule will combine with one RUBP. So 12 molecules of 6 carbon compound intermediate short-lived intermediate compounds are formed, which immediately not 12 molecules, 6 molecules, which immediately splits into 12 molecules of phosphoglyceric acid. phosphoglyceric acid टूट जाएंगे. ये 6 इंटू 6 36 है. ये 12 इंटू 3 दैट इज पीजीए में कितने मॉलिक्यूल है कार्बन के? 3. तो पीजीए में 3 मॉलिक्यूल है तो 3 इंटू 12 दैट इज 36. इसमें कितने मॉलिक्यूल थे? 6. इसमें कितने मॉलिक्यूल थे? 5. तो गेम इज वेरी इजी. इसमें कितने मॉलिक्यूल आए? 1 इंटू 6. So 6 into 5 30, 30 plus 3 36, 6 into 6 36, 12 into 3 is also. अब आया यहाँ पे एक नया स्टेप. यहाँ पे क्या देखेंगे हम? 12 मॉलिक्यूल and A, D, P, H plus H आएंगे. 12 मॉलिक्यूल ऑफ N, A, D, P, H plus H आएंगे. And 12 molecules of ATP भी दे देंगे. So when alcohol has addition of hydrogen, it changes into aldehyde. So हमारे पास क्या आ गया यहाँ पे? 12 molecules of 3 carbon compound phosphoglyceraldehyde. Or you can also say glyceraldehyde phosphate, PGAL. ठीक है, PGALdehyde के लिए AL. तो इसको कहेंगे ये 12 पागल आ गए हैं. Of the 12 pargals, 2 pargals runs out. 2 PGAL runs out, and they will make 1 molecule of ग्लूकोज. इफ यू रिमेम्बर यस्टरडे हमने बनाए थे ग्लूकोज को मॉलिक्यूल को स्प्लिट किया था ग्लाइकोलाइसिस एंड वी हैव गॉट एंड देयर वी हैव गॉट व्हाट वी हैव गॉट व्हाट यू कॉल ग्लिसरॉल्डेहाइड फॉस्फेट एंड डाइऑक्सिड एसीटोन फॉस्फेट. वो फॉस्फेट वाले थे, इमिनो फॉस्फेट वाले हैं. तो ये ठीक है. तो ये ग्लूकोज बना देंगे हम फॉस्फेट वाले. अब बचे हमारे पास 10 PGA 10 पागल यहाँ पर. All these 10 parallel, they will take the energy of 6 ATPs and they will change into 6 ADP, and this 10 into 3 30, 6 into 5 30 RUBP बनेगा. ठीक है, ये अंदर गए हाइड्रोजन इसमें बाहर गया और ये आ गया 12 NADP और 12 ATP गए और वो आ जाएंगे 12 ADP. सो नाउ यू सी मैंने उसको सबको 6 टाइम मल्टीप्लाई कर दिया. इन योर बुक इट इज 1 टाइम. सो यू सी यू हैड इनिशियल मॉलिक्यूल, फर्स्ट मॉलिक्यूल, एक्सेप्टर मॉलिक्यूल है ना. दैट प्राइमरी एक्सेप्टर कौन है? RUBP है. RUBP 1,5 बाइफॉस्फेट. ठीक है, रिबुलोज 1,5 बाइफॉस्फेट दैट इज टेकन 1 कार्बन डाइऑक्साइड प्लस 1 वाटर कार्बोक्सिलेशन हुआ. तो इस प्रोसेस को हम क्या कहते हैं? वी कॉल दिस एज ए कार्बोक्सिलेशन. When the carbon dioxide is attached, यहाँ पे क्या हुआ? हाइड्रोजन इज एडेड. सो व्हाट इज दिस? रिडक्शन. एडिशन ऑफ हाइड्रोजन इज रिडक्शन. NADPH2 से हाइड्रोजन अंदर चले गए, सो इट इज रिडक्शन. और ये क्या है? रिवर्सल है ना. तो इसको हम रिवर्सल कह सकते हैं या रीजनरेशन कह सकते हैं. सो क्वेश्चन कम्स ऑन द Calvin cycle. What are the 3 steps of the Calvin cycle? Carboxylation, reduction and regeneration. ठीक है. 3 स्टेप में आ गए, 3 स्टेप में। नाउ व्हाट इज द फर्स्ट प्रोडक्ट प्रोड्यूस्ड? फर्स्ट प्रोडक्ट प्रोड्यूस्ड इज 3 कार्बन। 3 कार्बन वाला सब्सटेंस बन रहा है पहला, ग्लूकोज नहीं बन रहा। It is not the glucose produced, it is the 3 carbon compound. So that is why it is called tricarboxylic acids cycle TCA, है ना। तो ये TCA है और TCA के अंदर क्या बन रहा है? पहला कंपाउंड जो बन रहा है वो 3 कार्बन वाला बन रहा है। इतना आ गया आपको समझ में? तो हमारा जो है मेजर पार्ट हो गया। इन टुमारोज क्लास वी विल फिनिश द चैप्टर। दैट्स ऑल चिल्ड्रन, थैंक यू।